Hello dear students, in this lecture we are going to talk about anatomy of dicot, monocot, root, stem and leaves. We will discuss one by one all the things in detail. First we are starting with the anatomy of dicot root. Dicot root. So in dicot root, if we make a diagram, we come to know that outermost layer is epidermis. First we try to understand if this is the dicot root, and if we take a cross section of this dicot root, and if we see, we can see this type of diagram. Out of this we are drawing only this section here. Okay, so this is showing the epidermis. Now over the root, and it is a epidermis of root, so over the root there is an absence of cuticle, but the root hair are present. So you can see here there is a presence of root hair. Now inner to epidermis we find a cortex, because hypodermis is absent in case of this dicot root. So we have a cortex. Now after cortex we see here, so after cortex we observe here endodermis. So this is endodermis cells. Okay, so this endodermis is present inner to the cortex, and these endodermal cells have a thickening of suberin which form a Casparian strips. Now inner to Casparian strips, we see a pericycle, and here we can represent this as a pericycle. So this is a pericycle. Now inner to pericycle we find the vascular bundles, and in vascular bundles also we find primary xylem, that is xylem, and radial arrangement. Now in xylem also, in root exarch condition is found, so proto xylem are towards the periphery. So in such way this primary xylem is located. So you can see here this is a primary xylem, and in primary xylem, which is made up of proto xylem and meta xylem, you can see this has a narrow cavity. And it is a proto xylem, whereas the inner side this is meta xylem. This is a cortex tissue. Inner to cortex we have a endo dermis. Okay, and inner to endodermis you find here this is pericycle. Now when we see this xylem and phloem, this metaxylem and protoxylem forming a primary xylem, and this xylem and phloem show radial arrangement. So here we observe the arrangement of phloem. Here we find the arrangement of phloem, and this phloem is primary phloem. This is a primary xylem. Now in between them, the cells which are present here, they which occupy the space, that are conjunctive tissue. That are conjunctive tissue. So these are the conjunctive tissue. So by seeing this, we come to know that there is epidermis, cortex, endodermis, a conjunctive tissue, and there is a presence of radial arrangement of this vascular tissue. So by seeing this diagram, we also know that they have a exarch arrangement of this protoxylem. So if we understand this and we write a points regarding to the Dicot root, then here cuticle is absent because these are the root. Epidermis is single layered. Okay, next to that we find a cortex, next find a hypodermis. That hypodermis is absent, which is found in case of the stem. Inner to hypodermis, cortex is there. Cortex is parenchymatous and made up of thin walled cells, so they can have intercellular spaces as well. Now inner to cortex we find a endodermis. Endodermis have Casparian strips, and these Casparian strips are impermeable to water. They are impermeable to water. This endodermis is present. Now inner to endodermis we find a pericycle. So now important points start with here, that pericycle of Dicot root give rise to lateral root, vascular cambium, and cork cambium. So this vascular cambium, cork cambium, and lateral root arise from the pericycle. How vascular cambium and cork cambium arise, that we will study in secondary growth part. Now inner to pericycle we find a vascular bundle. Now when we talk about a vascular bundle are radial arrangement. Vascular bundle that are xylem and phloem show the radial arrangement, and in radial arrangement also here we can see cambium is absent initially. Conjunctive tissue is present in between xylem and phloem, Next to that, if we talk about vascular bundle, they are 2 to 6 in number, means diarch to exarch condition is found. After this 2 to 6 vascular bundle, exarch arrangement of primary xylem. In this exarch arrangement of primary xylem, Proto Xylem is towards periphery and metaxylem is towards center. So such way they are arranged, and that is known as exarch arrangement. So this is about vascular bundle. Next to that there is a pith, and this is pith is small or absent, and it is, if it is small means it is not well developed. So this is the characteristic of particularly dicot root. But in dicot root, whatever the important points are with respect to pericycle, that you have to remember that it give rise to the particularly, here you can see vascular cambium, core cambium and lateral root. That is important one. Second, vascular bundles are important one. Radial arrangement is there, cambium is absent initially, conjunctive tissue is present, and 2 to 6 in number. This is important point. There is a vascular bundle which are 2 to 6 in number, and they have exarch arrangement of primary xylem. That is important with respect to the root. Okay. Now next to that we talk about a monocot root. Next is the anatomy of monocot root, and in monocot root also we are going to draw only this part of the cross section. Okay, when we draw this part of cross section, here also we find that there is a presence of epidermis. Now inner to epidermis, here also there is a presence of cortex. Inner to that we find a so this is the cortex. Now inner to cortex we find a endodermis. Now inner to this endodermis we find a pericycle. This is the cells of pericycle. Inner to pericycle we find the vascular bundles, and when we find the vascular bundles, and this red colored are the phloem, and these are the conjunctive tissue. And centrally located pith. So this is the xylem, a primary xylem. So if we label it, we come to know that they have a root hair, then there is a epidermis, this whole is a cortex region, then this is endodermis which has a Casparian strips, then this is a pericycle. Now pericycle of monocot root give rise to the lateral root only. They cannot form a core cambium or vascular cambium because cambium are the dividing tissue, and which this core cambium and vascular cambium help in secondary growth. But actually secondary growth or girth-wise growth does not occur in case of monocot. So there is a no need of formation of core cambium and vascular cambium. So because of this pericycle give rise to the lateral root only. Now inner to that we find Proto Xylem, then Meta Xylem, this is the primary xylem, then these red colors are primary phloem, they have conjunctive tissue and centrally located pith or medulla. Now when we talk about the characteristic and when we note down this characteristic, we come to know that they have epidermis, they have root hair, they have cortex, obvious cortex have intercellular space, cuticle is absent. There are common thing. What is the difference? The difference is important one, that pericycle in monocot root give rise to lateral root only. Then inner to pericycle, if we talk, that is about vascular bundles, then in vascular bundles they have radial arrangement, but with radial arrangement of vascular bundle are more than six, that is more than exarch vascular bundles are present. Okay, more than 6 or exarch vascular bundles are present. Radial arrangement is there. Here cambium is absent totally. Conjunctive tissue can be present. The important point is about the more than 6 vascular bundle. Now here also there is a exarch arrangement. In this monocot root, they are roots, so they have exarch arrangement, and they have a large and well developed teeth. Okay, so if we compare these points with respect to the dicot root, we come to know that the difference is the pericycle, difference in the vascular bundles. Okay, radial arrangement obviously there, but there is a difference in the number of vascular bundles, there is a difference in the pith, and that points make a difference between the dicot root and monocot root. Now after completion of this root, we come to the anatomy of the dicot stem, then we will talk about the monocot stem. Next we talk about the anatomy of dicot stem, that is a dicotyledon stem. Now when we see the dicotyledon stem, first we draw the cross section of this stem, and if we see the cross section of this stem, in this cross section of stem we see several layers up to the pericycle, and within pericycle we find this with the arrangement of vascular bundles. Okay, so many vascular bundles are arranged like this. Now if out of this we take a cross section of half of vascular bundles from both sides, and if we draw this in detail, then we come up with this type of diagram in which there is a outermost epidermis, and this epidermis give rise to the root hair. This is covered by a cuticle. This is covered by a cuticle. Now inner to this epidermis we find some cells of hypodermis. Some cells of hypodermis, and inner to hypodermis we find a cortex. So this is the cortex. Now inner to cortex we find a endodermis. Inner to endodermis we come up with the pericycle, and this pericycle is present in form of some of the patches. Okay, so like this pericycle is present. Now when we see inner to this pericycle we talk about vascular bundle, and in vascular bundle Dicot stem have a conjoint arrangement, so they have a phloem and xylem. So here you can see this is the phloem. Inner to phloem you will find here a cambium, that is a cork cambium, and inner to cork cambium we find a endark arrangement of this dark arrangement of this xylem. Okay, and inner to that we find a pericycle, a pith, sorry, inner to that we find a pith. So when you see this diagram you come to know that in between this pith elongates and that form medullary rays. Okay, so this way they are arranged. So here if we label it we find here there is a stem hair which are multicellular structures. Then you find here cuticle. Now cuticle is covering the epidermis. Now inner to epidermis we have a hypodermis, which is found absent in root but is present in stem. Then there is a cortex, general cortex. Then you will find a endodermis. Then this particular part represent pericycle. Then pericycle to inner to that there is a primary phloem. Then this is a cambium. Then inner to cambium find this is a metaxylem, and this is a Proto Xylem, and that is primary xylem, which is showing a endark arrangement. This is pith, and this are pith or medullary rays. These are pith or medullary rays. So now if we talk about this, then we come to that know that hypodermis is present in Dicot stem. Now we remember that this hypodermis which is present in Dicot stem, it is made up of collenchyma. So it is a collenchymatous. It is a collenchymatous. Now inner to hypodermis there is a presence of cortex, and thus cortex is parenchymatous. Okay, means it is made up of parenchyma cells. Then we talk about endodermis. Then this endodermis of particularly this Dicot stem is known as starch sheath. Why it is known as starch sheath? Because it has starch granules. Now then we talk about, when we talk about the another part of this, inner to endodermis there is a presence of pericycle, and pericycle semilunar or half moon shape. So it is present in form of semilunar or half moon shape, and this semilunar or half moon shape patches of sclerenchyma. So we remember the pericycle of Dicot stem is made up of dead cell, that is a sclerenchyma. Next to that we talk about vascular bundles. Now vascular bundles, this vascular bundles, if we talk about where a xylem, phloem and cambium is present, they are present in form of conjoint, collateral and open. So why conjoints? Because phloem is present outside, xylem is present inside, that is and that is collateral, and they are present in same bundle, so it is known as conjoint. And open means vascular cambium is present. Now when we talk about this vascular bundle, here in case of stem there is a endark arrangement of primary xylem, in which Proto Xylem are present towards pith or at center, and metaxylem are present towards periphery. So such type of arrangement is found in case of vascular bundle. This vascular bundle arrangement is known as a ring arrangement of vascular bundle. It is known as ring arrangement of vascular bundle. And due to the presence of cambium, this vascular bundle help in what? Secondary growth. Okay. Vascular bundle are of uniform size in case of Dicot stem. Then where they are wedge shaped, These are something a wedge shaped vessels arranged in rows, and phloem parenchyma is present in this. Phloem parenchyma is present in case of phloem, and medullary rays or pith rays are found between vascular bundles. Medullary rays are found between vascular bundles. And last we talk about pith. Then pith is made up of parenchymatous cells situated at the center. Situated at the center of stem. So this is the characteristic of which one? Dicot stem. So one of the important characteristics are what? Endodermis is known as starch sheath, semilunar patches of parenchyma in particularly pericycle, then conjoined collet and opal vascular bundles and dark arrangement. So these are the one of the importance with respect to the dicot stem. Now next is anatomy of monocot stem. Now when we study a monocot stem or the anatomy of monocot stem, then we draw a complete diagram of cross section. We draw a complete diagram of cross section. And in complete cross section, we first find the epidermal cells on the periphery. So this is the epidermis of monocot stem, which is a parenchymatous. Now inner to this we find a hypodermis. So you can see some cell arranged here like this that form hypodermis. This is hypodermis. So this is a hypodermis. Now inner to hypodermis we find a cortex, but cortex is not completely differentiated, so it is a not differentiated. So here cortex is absent and ground tissue is not completely different. So this is hypodermis, and ground tissue is not completely differentiated as scattered vascular bundle founds, and in this scattered vascular bundle we find a xylem and phloem, but this vascular bundle are larger in size towards the center, but they are smaller in size towards the periphery. So they are smaller in size towards the periphery. So if we see this diagram, these are the smaller vascular bundle and this is large sized vascular bundle. So when the smaller vascular bundle are present towards the periphery, they are large in number because they are small sized, so they can accommodate a more in number in a few area. But large size vascular bundle required a large area, so they are small or few in number. They are few in number. Okay, so this characteristic is found, and in this specifically when we talk about vascular bundles, the bundle sheath is found and that bundle sheath cover the all vascular bundles. So you can see here this red color covering, that is nothing but a represent what? Bundle sheath in and xylem and phloem. So this red color represent bundle sheath. So if we note down the characteristic based on this diagram of this monocot stem, we found that hypodermis is made of sclerenchyma cells. Now inner to that ground tissue is not differentiated into cortex and epidermis and cortex and endodermis, sorry. Now next to that vascular bundles. So these vascular bundles are scattered. So scattered vascular bundle are found in case of this monocot stem. Then large sized vascular bundle are present towards center, which are few in number but small sized vascular bundle are present towards periphery and they are large in number. Next, they have conjoint, collateral and closed vascular bundle. They are oval in shape and bundle sheath is present around vascular bundles. Okay, and phloem parenchyma is absent in phloem. Okay, and last, pith or medulla, that is absent. So these are the characteristic of monocot stem, having a scattered vascular bundle, then large size vascular bundle are present towards the center, small size towards the periphery, ground tissue is not differentiated. So this all characteristic make the understanding of the monocot stem anatomy. So after studying this, now we have dealt with the root anatomy, dicot root, monocot root, then dicot stem, monocot stem. So now the remaining part is dicot leaf and monocot leaf that we are going to study. So let's start with the dicot stem anatomy. Next we talk of anatomy of dorsiventral leaf or dicot leaf. So when we see the diagram of dorsiventral or dicot leaf, we come to know that, and if we take its cross section of leaf, we find that there is a presence of epidermis, below the upper epidermis you will find some of the cells which are columnar, column like cells we will find, which we call a pedicell mesophyll. They are a type of mesophills. Now we remember, in leaf the ground tissue is represented by mesophyll cells. There is no specific cells like cortex, endodermis, pericycle. This pith, this type of specific cells are not present in case of leaf. Only mesophyll cell represent a ground tissue. These all are the pedicell mesophyll. Here we will find a bundle sheath cells, closely packed bundle sheath cell covering is found here. Then they have another mesophyll which are free and have intracellular spaces, and this is lower epidermis. Now in lower epidermis we find such way a presence of stomata. Okay, such a way you will find arrangement of stomata. Now here in this epidermis is covered by a cuticle. So this is cuticle. This is adaxial epidermis. This is adaxial epidermis. Adaxial means upper epidermis. And these are the mesophyll cells which have a large number of chloroplasts. They have a large number of in this palisade mesophyll, and this palisade mesophyll are closely packed columnar, so intercellular spaces are absent. Next, this they have some less chloroplast, and this less chloroplast containing cells are known as spongy mesophyll. And within this spongy mesophyll we find intercellular spaces, so intercellular spaces are present. These are stomata, substomatal cavity, and this is lower epidermis known as adaxial epidermis. Okay, here these are xylem and these are phloem. So xylem is present towards the upper epidermis and phloem is present towards the lower epidermis, and they are covered by a specific layer of bundle sheath. Okay, so here what we understand from this anatomy of the particularly dicot leaf. So we understand this anatomy of dicot leaf, and we get to know that here epidermis covered by cuticle. Next, stomata are mostly present in lower epidermis. So this arrangement of stomata is known as hypostomatic. Okay, then mesophyll cell represent a ground tissue, and this mesophyll cell are of two types. First is palisade mesophyll. So in this palisade mesophyll they are columnar and they are closely packed, so intercellular spaces are absent. Whereas second type is spongy mesophyll. Spongy mesophyll have large intercellular spaces. Then next we talk about vascular bundle. So vascular bundle are present in midrib and veins. They are present in midrib and veins. They are conjoint, collateral and closed. So conjoint means xylem and phloem is present in a single bundle. Xylem is present towards upper epidermis and phloem is present towards inner epidermis. Okay, so that is the arrangement of conjoint collateral vascular bundle. And vascular bundle covered by bundle sheath, are covered by a bundle sheath. So that is the arrangement of anatomy of dicot or dorsiventral leaf. Now why they are known as dorsiventral leaf? Because on the lower surface there is more number of stomata. Dorsal and upper, dorsal means upper and ventrally lower, so dorsal and ventral surface are flattened, and the anatomy is different internally on the dorsal and ventral side as they have two different type of these cells or mesophils. So because of this, it is known as dorsiventral leaf. Now we talk about a monocot leaf or isobilateral leaf. Next we talk about anatomy of monocot or isobilateral leaf. Now when we see monocot or isobilateral leaf and we take a cross section of this, we find a diagram somewhat like this. These are mesophyll cell. Now in mesophyll cell, one of the point that you should remember that there is no division in type of mesophyll cell like palisade and spongy as we observed in Dicot Stem. Here only one type of mesophyll cell are observed, as you can see here, and they are closely pactive or closely associated with each other. These are bulliform cells. This is adaxial epidermis. These are mesophyll cell. This is abaxial epidermis. These are stomata. This is xylem, phloem and covering of bundle sheath. So now this is the structure of what? Monocot. Now monocot is known as isobilateral leaf, as we can say that number of stomata is equal on both surface, that is upper and lower epidermis both have a equal number of stomata. Then bulliform cell or motor cell, they are also named motor cell, are present in upper epidermis. These bulliform cells are large sized, they are large sized empty colorless cells. They are large sized empty colorless cells, and they help in rolling and flattening of leaf. They help in rolling and flattening of leaf. Next, mesophyll cell are not differentiated. Mesophyll cell are not differentiated, and vascular bundle are conjoint, collateral and closed. And the last point, bundle sheath is present around the vascular point. Be remember, cuticle is covering also on the upper and lower epidermis in the diagram. So at last you can see all the structure in case of diagram, and that is the structure of monocot leaves. So now till this we have discussed all the characteristic of dicot root, dicot stem, and dicot leaf, monocot leaf in this lecture. Okay, so now this is all about the anatomy of different leaf organs of different leaf part, different organs. In next lecture we will start with the secondary growth. Thank you.